What if artificial intelligence could design biology better than we can?
Maybe one day you'll have a little printer at home that can print your medicine, or you can print your food, I don't know, or on Mars, one day we'll live on Mars, according to some people.
Could algorithms learn the language of genes and rewrite it to make life-saving proteins more efficiently than nature itself?
AI is critical in re-engineering proteins, okay? And you see AlphaFold and all the novelty in this side. AI to be able to engineer new novel proteins. I think that AI will be wonderful to all of us. It will make the world a better place.
If that is possible, what does this mean for scientists, medicine, and the future of biotechnology? Well, in today's episode, I'm speaking with Eran Miller, co-founder and CBO of MNDL Bio, a company using AI to revolutionize how we optimize gene expression and produce recombinant proteins. Together, we will explore how deep learning is transforming synthetic biology and what democratizing biotech really means, and whether AI will replace or simply empower human scientists. Have fun and enjoy our conversation.
Daily dose of honest science.
Hi guys, welcome back to Daily Dose of Honest Science, your go-to podcast for evidence-based conversation in biomedical sciences. I'm your host, Dr. Chandni Kumar. Today, we are diving into a topic that's shaping the future of biotechnology, the democratization of synthetic biology. My guest today is someone who sits right at the intersection of innovation, sustainability, and science entrepreneurship. Joining me is Eran Miller, the co-founder and chief business officer at MNDL Bio, a Tel Aviv-based company pioneering solutions to make synthetic biology tools more accessible, affordable, and impactful. MNDL Bio is transforming food and biotech production with advanced computational and AI-powered platforms that optimize gene expression. In simple terms, they're helping companies to produce essential proteins in living cells faster, more efficiently, and more sustainable than ever before. Well, that's a task I'm well aware of and with many pitfalls. Their technology eliminates much of the traditional trial and error that wastes time and money in protein engineering. Instead, they use data-driven algorithms to increase yields, reduce costs, and accelerate developmental timelines. Founded won more than 20 years of pioneering research by Professor Tamir Tulla at Tel Aviv University, MNDL Bio's platform has already proven its value through collaborations with leading biotech and food tech companies. And in 2023, they were awarded the Caller Startup Competition Prize for their breakthrough innovations. Eran has over a decade of international business development experience and a solid background in R&D, from leading agriculture projects across Africa to driving technologies ventures in Asia and Pacific. He is passionate about building bridges between science, technology, and sustainable development, and is now focused on preparing for a future where, he says, biology speaks to everyone. A big warm welcome and big applause for Eran Miller. Hi, Eran. Thank you so much for coming to the podcast.
Thank you very much. What a wonderful introduction. Thank you so much. Amazing to hear it from you.
Yeah, I was really excited to talk about this because usually, I have people on the podcast who are academics. Now I was really thrilled to have someone who is in biotech and doing, having a company. So we get right away, Eran. So let's start very from the beginning. What were the key moments in your early life that shaped you actually to go into technology and innovation?
Yeah, I think I've always come from an entrepreneurship mindset. I like to see things being built. I like to work with my hands, building things and making things happen. So I traveled a lot, like you said in your introduction. I traveled really all over the world, bringing technologies from one place to another, and creating projects and building stuff.
Mm-hmm.
And I think that having seen many different things and challenges, and working with people, made me understand that let's say entrepreneurship is what I like to do. Science and technology, bridging, like you said, bridging science and commercial objectives is what makes me happy when waking up in the morning, and that's what I'm doing for many years already.
Oh, that's so cool. I know that someone else who joined the podcast some episodes before, Dr. Eli Gottlieb, has kind of the same passion. So he came from the academics, but he was like, I want to have sleep technologies that really help people. And that's why, he joined a company and is now doing there the lead research. And I'm so excited to talk about this. From your background, you actually studied electrical and electronics engineering first, and then you actually go into the world of executive MBA. So, can you explain why this subject and how did you transition actually?
I'll tell you like this. I had, like, my personal career is divided into two. I had about 12 years, something like that, in pure R&D, working with very complex software. I'm an electrical engineer by profession, but I worked on many ventures and projects in companies all over the world, in basically in Asia, in Europe, and in Israel. And I worked in software and in hardware development for about 12 years of my first half of my career. And the second half of my career, I decided to switch from R&D more to business, and and I went into food and agri-tech, more business, commercial business development, sales roles, creating ventures. And to do this transition more, let's say, more smoothly, I went into school after a few years of R&D work. I went into school to learn some of the basics that you need for better doing this transition. So economics and financing, and how to run a company, and how to establish a company, and legal aspects, and so on. So
And how difficult was it for you to actually do the transition? Because when you do R&D, you're actually not having a good foundation for business, right? So how much of that was kind of new to you?
So I'll tell you, I actually grew up in a family of entrepreneurs. So I saw it firsthand, and it was always present at home. And I always knew, even when I was on the bench developing some software or fixing bugs or something like that, I always knew that sooner or later, I'll move from pure R&D or managing R&D projects into the business side.
Oh, that's really cool. So you already had like a little of the foundation and then you decided to go deeper into it, which is really nice. My audience is quite diverse, and I would like to give you the opportunity to actually explain what your current role as Chief Business Officer actually is at the MNDL Bio.
Okay. When you a company, the most important thing in a company, or one of the most important things in the company is having customers, and even more
Yeah.
important, having paying customers. Having paying customers with a deep tech company like Mendel in a competitive market and a difficult landscape, like I guess we'll talk a bit later,
Mhm.
is challenging, because you need to create the right partnerships, you need to avoid the risks, you need to spend your resources, and they are limited. In a startup company, the resources, in every company, the resources are limited, but in a startup, it's really your every day you need to choose where you put your resources and your time, not only money, also your time and the people, the team's time. So having a mindset, a business mindset, and an understanding of the needs of the market and how we can tackle these needs with the scientific development that we have, that's the chief business officer role. So translate the R&D into actually, at the end of the day, revenues for the company, and recurring revenues, and a long duration revenues.
And what are currently the biggest challenges, let's say, that you have to face? Can you give like an example? What is currently?
Yeah, one of the challenge, what we do, our product, let's say, is sold to scientists. Okay,
Yeah.
one of the characteristics that you need to have to be a good scientist is to be doubtful, to doubt and to question everything, right? Not to take anything as
True.
is. So
Yeah.
when we come to a customer this customer is a scientist, and we tell him, look what our product can do for you, and look what we did for others, and so on. He asks questions, and he wants to see validations and proofs, and so on and so forth. And again, we go back to us being a startup company. A startup company that is based, like you said in the introduction, on 20 years of research, but still a startup company. So it's a challenge that we are overcoming. And we have more and more validations and more customers and partnerships, strong partnerships that we're creating. So this part is getting a bit smoother, but we're getting ready for the next challenge. But that right now, in the past, I would say 6 months or so, if I need to pinpoint the challenge, the business challenge of the company, would be that.
So it's like the problem having the scientists to tell them like it's actually a good product, and tell them like you can use it. It's actually really valuable for you. And now you have like a portfolio where you can say, well, XYZ project has shown that it works. Now it's making your life a bit easier to tell the scientist, hey, it's actually a good product, so you should invest.
That's correct. If you're a scientist working on mammalian cells and you are producing a certain antibody, so you want to see another project with the same cell producing the same antibody.
Yeah.
But if you don't have this in your portfolio, you need to be a
It
little
will be
bit.
difficult.
It will be difficult.
In the introduction, I already said that you had a lot of stations in your life across Africa, Asia, and Israel, of course, and working on projects that had to do with agriculture, food, and technology. What would you say the international experience that you gained were actually now helpful to you for the innovation in the biotech? So how much of these experiences are actually now coming into play in your role that you're doing and for MNDL Bio?
It's important in actually every position. Every position that you have, I think it's important to be able to accommodate yourself to different cultures, especially today that it's so easy. We speak over. You are in Germany, I'm in Israel.
Yeah,
There is
exactly.
absolutely
Yes.
like the geographical barrier completely vanished a few years ago. So especially in this new environment, you have to be able to speak and to work with different cultures. Look, we have 2 employees in India. We have, of course, our Israeli office. We have 2 employees in the US. and 2 in Portugal. We are all over, although we're a small company, but still,
Yes.
you
Yeah.
need to you need to accommodate both in cultures and in working hours. Not always easy.
And how much of this experience that you gained over the time shapes actually also the product that you are making? So, are you maybe tailoring also sometimes your product to specific needs in specific countries, or would you say it's a global established product that you could use anywhere? Or do you have like specific niches that say, well, I don't know, in Portugal, we need to have it a bit tweaked because we are working actually on something different than someone is working in the US?
I'll tell you, I think that one of the main, um, a very important thing in understanding your market and in tailoring maybe your product to different markets is to listen. To
Mm.
listen is not always easy because
Yeah.
you have your science in mind, you know what you can do, and on the other side you have a customer. So do you try to sell him something, or do you listen to his problems, to his problems
Mm.
which are related to what you do? So you have to listen, and also listening is related to culture and is related to being able to work all over the world. And it's a big advantage to be a global or a multicultural company like us, because
Mm.
then you have boots on the ground in different locations. But I
Mm.
think that even when speaking, and again, it's not important if you are doing biotech or you are selling clothes. Listening to your customers and your partners and your experts, and being able to translate what you hear into a product and to actual things that you are able to provide. I think that's a key that creates opportunities for the company.
Hmm. And let's say, how long is your consultation sometimes? So, like when you have a new customer, depending on the needs, I guess it takes a lot of time to actually listen and then make a product that maybe suits them, right? So,
Sometimes you come to a customer, you explain what you do, he tells you what's his pain. There is no interaction, there is no intersection at all, and
yeah.
then okay, you start speaking with him again. You
Mm.
understand that maybe we can do something to. It can be between 5 minutes. Okay,
Hmm.
you have this challenge. You want to express this protein. You're working in this bacteria. Okay, tack, tack. And that's it. It's very quick and easy. And
Mm.
sometimes it takes 6 months because he
Mm.
is in the middle of a project, or he doesn't need, or his needs are something that we haven't yet developed, but we decided to
Mhm.
develop, and
Mhm.
and so on. So
Mm.
it really changes from one case
Mm.
to another. But
And the products that you have right now, are they all based on the problems that scientists had at one point, and then you said, I develop, or you developed the solution for that? Or how do you actually get at the front of it and be like, okay, we want to be a leader in this AI driven platform? So how do you sometimes know what kind of challenges the scientists have if they don't talk to you? So how do you get to know that?
So we are lucky to be. We have 3 co-founders. One of them, like you explained, is Professor Tuller from the
Yeah.
university, and we are lucky enough that Professor Tuller is a visionary. Okay,
Mhm.
he's clearly a visionary. 20 years ago, when synthetic biology, nobody knew what synthetic biology is. I don't even know if the word tech bio
Mhm.
existed. Maybe yes, I don't know. But he understood that there is one bottleneck of a few companies, and back then it was not a widespread
Mhm. Yeah.
economy, bioeconomy like today, but there
Mm.
was there was one bottleneck that almost nobody, or maybe nobody, tackles. It's how to
Mm.
engineer DNA.
Yeah.
And it was way before any company or any potential customer told him, look, we have a problem. People didn't understand even that it's a problem. It was
Mm.
part of the workflow of the company. It still is, by the way. But
Yeah.
companies wanted to reach a certain objective. They had to spend 6 months on designing their DNA or the recombinant DNA. So they spent these 6 months. Sometimes it worked, sometimes it didn't work. Sometimes they spent millions or tens of millions of dollars and years, sometimes not always months. And he understood that there is this bottleneck. Is like we said before, he's a scientist. He argues with everything, and he needs to prove and understand everything. So having understood this, he went into modeling what's called the gene expression and modeling the living organisms and try to understand how they work in order to be able to re-engineer them with engineering tools, like I learned at school 25 years ago in electrical engineering school.
Yeah. So this is and the whole field itself, if you look at all the publications that came out, are actually designed to get more expression and more, that you can actually study the protein that you're interested in. And that's one of my big works that I do in the lab, because I'm a biochemist. So I know the pain of certain things not working and then trying again. What kind of solubility tags could you add on it? What kind of different buffers could you use? So all these kind of things, right? In the end, you end up sometimes with insoluble proteins, so you have to denature them and refold them and all these kind of things. So it's quite impressive that you have taken the lead to actually prove that there is a pipeline that you can have to make your life easier, right? One thing that I'm quite interested in, and that's why also having the talk with you is, since my audience is a bit diverse, so we have people who are from academics, but also industry, medical professionals, but also like normal people who are just interested in science. From your perspective, what can a scientist, I actually can learn from you, right? So usually this is now coming up. Universities try to make more startups, right? So scientists become actually entrepreneurs. But my question is, what kind of foundation do I need to be a good and successful entrepreneur? Because as a scientific background, okay, I have that. But as an entrepreneur, I think these are completely different skills. And it would be really nice if you could walk us through like what you have actually learned over the time.
I think that first, to create a successful venture is all about people. Okay, it's not a one person, one man show. It's not that one person leads the whole. There is one leader, but he needs a set of skills which nobody has all the set of skills. Of course, the scientific, we are in deep tech. You need the science, and our chief scientist, CSO, chief scientist officer, which is Professor Tuller. His role is to look, let's say, 6 months to 5 years ahead, and to tell us, okay, we need to go that direction. We need to tackle this challenge, or think about a solution for that, or let's things like that. But behind, and it is required, because we're not a one shot. It's a short duration strategy. So we need this part. It's a minimal requirement. But after that, as you say, you need other layers as well. You need the engineering layer. Engineering layer will take. That's my personal background. Will take let's say a scientific challenge or a very complex situation or condition, and we'll build the blocks of, okay, let's understand how we solve this problem, or what do we have in hand, so we can bridge a certain gap. That's the second layer. Another layer that you need is everything related to the business side, to what do you sell, to whom, why, at what price, what your competitor is doing, and so on and so forth. And then, of course, you have your first partners who are the financing partners, the the VCs who invest in the startup. So you have a few layers, and that's why we are three co-founders, each one with his own background, experience and background. Yeah.
And thank you so much for sharing, because for me, always like the business side is something that I think I could imagine to learn it, but I think it would be very stressful for me to also have that in mind. I rather focus on the research, but it's really cool that you have so many people and with different expertise that you can actually gather together and make the best product out of it. Maybe coming shortly back to a product. So what was the longest time that you needed to make actually a product because you faced something that was easy in the research, but actually not so easy in the turnover, like in engineering part or even in the business part? Because sometimes things are too expensive, so you cannot really give that. So do you have like a product where you said, that took a bit too long than we actually anticipate because we ran into XYZ problems?
Yeah, I'll tell you. It's a good question. I'll tell you. If I think I have a few. If I think I have a few examples, but one of them, and I'm sure it will be familiar to some of the people who listen. We're going to AI. So we have a toolbox which is full of different algorithmic, let's say, approaches. Some are AI, LLM, neural networks, biophysical models, many things that we have in our toolbox. But one of the things that we're improving now is LLM, is being able to understand the language of DNA and understanding how to write it, how to write it from scratch. There is a lot of effort all over the world about that. Taking LLM into the world of what we do, with genomic data, and it takes an unknown time to reach a certain point, to reach a certain goal. Why it's unknown? Because it's research. So you can expect a certain milestone to take 2 or 3 weeks, where it could take a month and a half. You can, the opposite can also happen. It doesn't happen often, but it could. After that, you need to validate things. And as you know, biology and validation is, on one hand, it's a must. On the second hand, you can do an experiment 1 week. The second week, you do exactly the same experiment, and you have something else because the temperature went up by 0.2 degrees.
Yeah.
So there are always, you always, it's a startup, it's a roller coaster. So so you need to be ready with a second plan, a third plan, and fourth option. But definitely, when implementing AI, we have a few directions. We put ourselves a few programs to develop AI with different models and different approaches, and LLM and small language models. So we have a few approaches, which is very difficult to estimate in time, and again resources, like we spoke in the beginning, and to put a number here. You can put a number, and then 2 weeks later you see, I forgot this, or this doesn't work on that. So it's a challenge in R&D. In managing R&D perspective, it's a challenge.
That's really interesting to know that even in company settings, we have these problems that we don't overcome in academia or industry. It's always vice versa. But I guess in industry, the thing is you need to be keep the money together, right? You don't have like XYZ options to do. So you always need to have that in mind, right? What I really like is you have described yourself as a very passionate about sustainability and impact. So I would like to know, what does this actually mean for you personally, and especially what do you define impact in your work now?
So this too, by the way, is something that I grew up personally. I grew up on this on the on this notion of sustainability and economic impact and impact in general. What we do in Mendel, actually, this whole world of biology, at the end of the day, is using or engineering nature or engineering living organism to make good things, to feed people, to produce medicine, to produce leather without animals, to produce milk without cows, and to do good things. In order for this to happen, and to go to the supermarket and see meat which was not where killing an animal was not part of the process,
Yeah.
you need to lower the cost of production. One of the and we go back to the bottleneck. At the end of the day, it's a business. A person who sees a steak for $2,000 will most probably not buy it.
Yeah.
And one of the bottlenecks is designing the DNA. So, and we are really in the heart of it at Mendel Bio. So seeing it firsthand, to see these things, where we were 15 years ago in the research in the university, where we are 2 years ago when we started Mendel Bio, and where we are today, and where we plan to be in a year or two, it's really amazing. It's fascinating and it's exciting.
Yeah. Yeah. And also having the AI component now in there as a very prominent part in your workflow is something that gives you like a hidden layer that you wouldn't have maybe gotten to it if you would do it manually, right? So sometimes AI finds patterns that humans, for whatever reason, don't find, right? And that's really cool to see like how you can leverage your whole product to another level. Just very shortly, what kind of products did you do, especially now with food? Because you mentioned it, making milk and so on. Just very briefly, where your tool actually helped to get a product much better for food, because we know we all have the crisis of many people around the world, right?
Yeah.
The population is rising. The food is getting, unfortunately, less because of all the environmental changes that we have. And many crops are dying because there's not a safety mechanism to actually grow them, because the soil is not having nutrients enough, or too much sun is actually damaging them. So what kind of foods actually you help to produce, so we actually get into that direction that we are helping the global population?
So I can tell you two examples. One is we work with a startup company who is developing an enzyme which aims at turning sugar, like glucose, to another type of sugar which is healthier. Now, their product is the enzyme itself, but it's the food industry, so it again needs to be cost effective. So they wanted to lower the cost of production of their enzyme. So so we work with these guys. That's one example. Another example I can give you is one of the major factors of cost for the alternative proteins, so cell-based meat, is growth factors.
Yeah.
Growth factors are expensive. Some of them are extremely crazy expensive, like millions of dollars per gram. Yes,
Yeah.
it's.
Yeah.
So we work with companies that produce these growth factors, again in the same objective: increasing the yield of production and lowering the cost of production for these growth factors, which eventually they will lower the cost of the meat which is produced.
Are these then the synthetic ones? Because I know some companies offer for cell culture, for instance, synthetic FBS. So it would be interesting to know if it's something in that
No,
range.
we work with what our customers they produce. No, for example, FGF, growth factors. It's not synthetic. It is not produced by the cow. It is produced by a bacteria or by a yeast or
Okay.
by a plant. It's exactly the same. Molecularly speaking, it's exactly the same growth factor. So it's the same chain of amino acids.
Okay. Now I got it. Okay. Really cool, and thank you for these insights. Finally, I would like to go to the next section, but before we do that, I would like to know from you, what are your personal goals for the next few years, and maybe what kind of changes you want to see or drive, how we use biology to solve global problems.
I think we are in an era. I'll tell you like that. 3 years ago, Professor Tuller and told me that, and he was the first one to tell me that, and since then it fascinates me that nature is the best factory that exists, right?
Yeah.
We, in our body, we have the capability to produce 25,000 different products, really 25,000, okay, because we
Yeah.
have plus minus 25,000 genes. And a plant can produce also tens of thousands. So nature is the best factory. We are in the beginning of what Jensen Huang from NVIDIA calls it the bio-revolution. It's the biggest revolution ever, and so on.
Yeah.
But we're in the beginning. I think biology and using biology or using nature or engineering nature to do good things will only is here and is here to stay and will be all around us. I have I have 3 kids. They are young. They're not yet choosing what they want to do later on in their life, but
Yeah.
no doubt that whether they choose to go into life science or not, they will. It will be the world will be different. The biology will be all around us.
Yeah. So you are trying to actually sustain the biology, make the best out of it, and keep the fascinating science behind it, right? So that would be your main goal, which is really beautiful. Let's hop onto MDL's mission, where AI meets biology. And what I would like to know is, so you said it's been now 2 years. You were co-founder of it, but my question is, how did actually the idea come to do this, to do the startup, right? What was the moment where you all sit together and be like, "Hey, we just need to do this company now"?
So first, it's Mendel, not MNDL. Mendel, like Gregor Mendel. It's based
Oh,
on
okay. Now I got it. Okay.
yeah, like Tesla, and yes.
Okay.
So, like I told you, 3 years ago, we met Tamir Tuller, Professor Tuller. Steve, my co-founder, we're friends from Belgium. We grew up in Belgium, both of us. He comes from the food tech industry. I come from agri and food tech, and we wanted to solve something around alternative proteins. Actually, that was our first choice. That was more than 3 years ago, maybe about 3 years ago. When we saw Tamir, Professor Tuller, he explained to us that he has a good technology that can solve many things around alternative proteins, but the world that the commercial or the business opportunity that he is solving is much larger than alternative proteins. It's medicine, cosmetics, biomaterial, and so on. It took us months to understand the concept, to understand what, like you asked before. He is a scientist. He's a scientist who had great science. He also, by the way, he also worked a lot with customers, with collaborations. But our goal was to scale up this technology. So until 2 years ago, he did, let's say, his in the academic setup. They did a few dozens of projects, but there are thousands of proteins every year. So this moment, when we met him, when we understood that scientifically his technology is mature enough, that he has proof of concept, and we built first an initial business model. When all of it took form and shape, then we understood that we wanted to create a company around that, and then we raised money also.
Oh, okay, that's really interesting. And MNDL Bio has this clear mission, of course, to make advanced synthetic biology tools more accessible and affordable, as you said. So when you look at this landscape of biotech companies that have similar products, what was like let's say, the initial nudge for yourself that be like, okay, we need to make this product right away? So because there are other companies, of course, they also have kind of like similar algorithms, let's say. But what would you say makes MNDL Bio so unique?
First,
If you had to pitch now your business.
we have a few things. One is, there are not so many. I don't know any actually, or not many, I'd say, company that are able to combine between, let's say, AI, the world of AI, and biology-based algorithms,
That's
biophysical.
true.
And they are not. It's a capability that doesn't exist in most of the companies. Maybe it exists in some of the research labs,
Yeah.
but in commercial companies, I don't think that it exists on a commercial, for sure, not on a commercial scale. And there are few reasons for that. DNA, engineering DNA, you need to understand that if you give me, as you know, you're a biochemist, if you choose a specific protein, you have
Yeah.
almost an endless amount of ways to engineer DNA. Yes, you have
Yeah.
codons, you have an average 3 codons for every amino acid. Let's say you have, for the average protein, you have one with 200 zeros after the one options to re-engineer
Yeah.
the DNA.
Yeah.
So you cannot have enough data to train an AI model that will predict in such an endless amount of possibilities. Also, expression data is not something that is easily found.
Yeah. That's true.
It's either proprietary or not interesting enough, so it's not kept.
Yeah.
And so there is DNA data, there is transcriptomic data that you can find. Also, not for all of the host organisms, but it
Yeah.
exists. And our ability to combine these two worlds, this AI capability and synthetic biology or biotechnology world, that's our core value. In addition to the fact that we have 20 years of experience working on
Yeah.
collaborations, so we have
Yeah.
our own proprietary data as well.
I just give you an example from my research right now. So I'm working with a protein that is highly disordered in the middle. So I have N-terminally, it's well defined, and C-terminally, but in the middle it has like this very weird structure, let's say. So disordered means that, for the audience, that it doesn't have a really well defined structure, and that already makes it, for like AlphaFold, really hard to predict the structure, right? So I would like to know, how does your AI tackle this problem, right? So, you know, basically from the sequence, you could actually extract the information how it might be folded, right? So how do you actually juggle around with these problematic proteins, let's say? And also one thing, my protein seems to be on the transcriptomic level, so on the mRNA level, to be expressed good. But in the protein level, when I do my mass spec stuff, it's not there. So how do you actually juggle around with these kind of different proteins?
It's not there because it's not well folded, or it's not there, it's not soluble, or you don't know?
It's not well. It's. I think it's so low expressed that you cannot detect it. If I overexpress it in my cells, so I work in HeLa, then you see it, but not if you have the normal status of the cell, which is quite interesting to know.
I'll start with a disclaimer. You cannot solve any problem by engineering DNA. Okay? Sometimes it's you have a toxic protein, or you
Yes.
have proteases, or you have, I don't know, pH levels that are not allowing folding, or I don't know, whatever. So we are not able to solve any non-solubility, low solubility challenge. Having said that, we are looking, when we develop algorithms, we are looking at the whole chain, the whole gene expression, the different phases of the gene expression, starting with transcription initiation and all the way up to co-translational folding. The protein that starts already to be folded when it's still attached to the ribosome.
Exactly.
And when we speak and we understand, it goes back to listening to your customers. When you listen and you understand, you ask the questions. Sometimes we can find locations or things that we can try to improve. For example, improve the rate of translation of the ribosome. Oftentimes we see that it affects a lot the folding of the protein. Sometimes it solves problems, sometimes it doesn't. Sometimes using the right linkers or changing linkers solves such problems, sometimes it doesn't. So really we need to go into, I don't know exactly the protein that you're using. I'm also I'm by profession, as you know, I'm good in software and hardware. I'm trying to improve my biology skills, but
so in my case, I managed to get it recombinantly, but I was just puzzled that inside the cell, when I study, like if I want to check what is in vivo happening inside the cell, then it seems to be very low expressed for a good reason that I figured out. So hopefully I have that paper out soon.
No, well done.
It has so biology is not always very straightforward. Some proteins are well expressed because we need them in the cell to do many tasks, but some proteins are very low expressed because they have very defined tasks. And this protein has been already shown to be, if it's overexpressed, promoting a certain cancer type. So that is already hinting towards an overexpression of a certain protein is not good inside the cell because it would promote cancer. So it's really interesting to know. And also you're kind of helping to understand certain aspects, because sometimes, as I said, you want to study the function of this protein inside the cell, or in my case, also biochemically by doing certain experiments to figure out what is the binding partner, what is how stable is it? Is it binding to DNA? Is it maybe binding to RNA? All these kind of things to characterize what it's actually doing inside the cell. And getting it into the tube is sometimes really, daunting because it's not easy. So your platform is quite cool to also get that biological data that is existing for certain proteins already also in, and it's really, really cool to see how you actually can improve that. And One thing is to also keep the cellular fitness. So sometimes you have, I think,
Yeah.
customers who actually want to express it not in E. coli or in yeast. They want to do it actually in the mammalian system. So how does Mendel Bio's AI platform help to actually secure the cellular fitness? So what is actually like kind of the important information that you tell the customer, be like, okay, you need to be aware of this and this topic. So how do you do that?
Um, the fitness of the cell is definitely something that we don't want to harm.
Mm,
Again, it's not always things that we can shape or impact through
mm,
our tools.
mm,
Um, but as we know, if you overexpress, like you just said, if you overexpress a certain protein which can cause toxicity or which has negative
mm,
effect of some sort,
mm, mm. Yeah.
it's overpopulating the cell or harming it. So you need to be aware of what you're
Yeah.
doing. That's why we have molecular biologists in the team. We're
Mm,
mainly a computational company, but we need to understand the molecular biology
exactly.
side of it.
Mm.
Um, yeah, I'm not sure that I completely answered your questions,
No, I mean,
but
so you have people who actually take care of this part and tell you like, or tell the customer that some proteins might not be good to overexpress inside the cell because whatever can happen, that it's toxic or and so on, right?
yeah,
So
we
that is
with
also.
men. Yeah, actually, with many customers, we work really on long. It's really long-term collaboration or partnership.
Mm,
We
mm,
start with one project and we discuss and we change and we improve internally. We improve. So 6 months later, we can work again and provide
yeah.
improved DNA. And so it's really a,
Mm,
a, a collaboration. We learn a lot from these collaborations, and we
mhm,
also provide value to our customers.
mhm, mm. Um, to your platform, I actually chose two interesting publications that you were linking on your website. So one was AI-directed gene fusion prolongs the evolutionary half-life of synthetic gene circuits, and the other one was a tool for CRISPR-Cas9 sgRNA evaluation based on computational models of gene expression. So these two examples were quite impressive for me. That your AI tool helped actually to get more specific insights. And I would have the question, so could you maybe share like the walkthrough in these two publications? So what did Mendel Bio actually really did there that it enhanced our overall understanding in these two topics?
So the first one is actually a very nice capability that we have. We also patented. We have a patent on it.
Mm-hmm.
Basically, it's improving the genetic stability of a gene of interest. So, as when you have a lot of microbes inside a tube or a bioreactor, the fittest will stay the longest. And the gene of
Yeah.
interest, which the scientist or the user is interested to produce, let's say it's a meat, the growth factor or something that has nothing to do with this microbe, doesn't really interest the microbe. And to the contrary, it lowers its growth. It takes energy for the growth of this microbe and so on. So all of the mutations that remove this gene of interest and are not producing this
Mm-hmm.
gene of interest will grow faster than the ones who are producing. And then after a few hours sometimes, or a few days sometimes, your tube will have mainly cells that do not produce your gene of interest. So how do you overcome
Yeah.
this? There are a few ideas to overcome it, but what we did, and we proved with a few validations, that it works is, it sounds simple, but the actual doing it is
Yeah.
complicated. We
Yeah.
fused two genes together. So we took the gene of interest, we fused the gene of interest to an essential gene, which is not too much expressed in the cell. Okay, so the essential gene is basically a gene that the cell needs to survive.
Yeah.
And when we fuse them together, we glue them together. Sometimes
Mm.
the ribosome would produce only the gene of interest. Sometimes it will go, it's called a leaky stop codon. So sometimes it goes and produces both of the proteins, so the gene of interest
Yeah.
and also the essential gene. There is
Yeah.
a linker in between which is broken when going out of the cell, and so you have this gene of interest and also the essential gene. This makes and this created a dependencies and a life dependencies
Mm.
on the cell,
Mm.
keeping this structure. And what
Mm.
we
Mm.
showed is that the genetic stability is improved by 100-folds, or really something very, very
Mm.
large in some
Yeah.
cases. So this is the first publication that you mentioned. It's very interesting work.
It's a really cool advancement because
Yeah,
usually when you think of transfecting cells, so for the audience, usually you have a plasmid that you introduce into the cell and tell the cell actually to do the protein, but the cell, of course, has mechanism to actually degrade that whole
correct.
the whole DNA again. And so you're only limited by time to actually have this like a small time window where you can study the protein. And your system actually now helps to study the protein a bit more advanced, because if you have 100-fold better stability of the DNA, you can actually study it for much, much longer because it's not going to be degraded. You don't have to be very quick. You actually can study it over a longer time, which is very important, right? So it's a really cool advancement. But I also would like to go into the CRISPR evolution-based computational model, because I was thinking like CRISPR has been around for a while, and why is it important to have AI to actually make CRISPR even better? So maybe you can go a bit more into that.
So I'll stay on vague here, okay? A little bit vague,
Mm.
but basically, yes, we know CRISPR, but CRISPR is being around not for such a long time, for maybe 10 years.
Yeah,
Commercially, it's even
commercially, yes,
less.
exactly. So people who study CRISPR, they know CRISPR a bit longer, right? But
Yeah, but
for commercial purposes, yes, that's true.
um, but CRISPR first is some downsides.
Mm.
First, it's very expensive. Producing the protein related to CRISPR, so Cas9 and the other proteins, is expensive. Uh, I believe it is not always specific enough or enough uh, specific or correct, or not correct,
Mm.
specific enough. And so we invested in it's actually Professor Tuller's lab researched a way to design guide RNAs and to a better design of this whole system
Mhm.
of CRISPR, CRISPR editing, and that's what the paper is about.
Mm. And would you say that this help that you did with your algorithm, because there's also a lot coming with CRISPR trials, right, to make gene editing possible for disease eradication or disease mitigation, right? So would you
Yes.
think that in some future you could use your algorithm to actually make more precise editing inside the cell?
It's definitely this is exactly the direction. Yes, of course, this is
Mhm.
exactly the direction. But you need also not only precise, you need it also to be cost effective. Not because these things, to develop a new drug costs billions of dollars.
Yeah.
And if the CRISPR-based drugs are not commercialized yet,
Yes. Yeah.
so we need also not only to be more precise, we also need to
Mm.
take the cost down. And
Mm.
yes,
Mm-hmm.
definitely, we see ourselves as in Mendel very active in these two directions, creating better CRISPR, but more precise tools by
Mm.
engineering guide
Mm.
RNAs or
Mm.
engineering the proteins around it,
Mm.
and also lowering the costs.
Mm. Are you? Can you share maybe if you're already working with customers together that are using your AI tool for clinical purposes?
We are. I'm not. I cannot share too much
No, no,
details.
but I mean, you are involved in also like clinical trials using your AI platform to make, let's say, therapies better.
Yes,
That's really, really cool.
yeah,
So, as you can see, guys, it's already happening. We are,
definitely it's happening.
yeah, at the forefront. So, if you're not excited, I don't know what to say. So maybe on that note, so one thing that really stands out for Mendel Bio is now that you integrate the biological data, right? So that you say the biology is important to understand for the algorithm what to do. But me as a scientist, I'm of course saying, how does the AI can learn from different biological contexts? Because you're working with many kind of different collaborators, right? They are not always specific in one field, right? They work in, I don't know, RNA splicing. The other one is working in HIV. The third one is working in whatever. So how do you ensure that the AI actually gets the biological context correct?
That's the beauty of our team. I must tell you. It started with Professor Tuller. Okay, Professor Tuller has about 40 scientists and researchers and students in his lab. They work together for, as you said, 20 years or something like that. And they come. By the way, Professor Tuller has 8 academic degrees, combining
Mm,
both the computational side and the life science side.
wow.
And
Okay.
and that's the beauty of the team. We have both. We have in these 40 people, we have uh, uh, physics students, and we have electrical
Mhm.
engineers and software engineers and
Mm,
mathematicians, but we also have physicians, so doctors. We have biologists, we have biotechnologists, we have bioinformatics, so we have chemical engineers. So it's really a mix of knowledge that you need to have to bring
yeah.
such a holistic approach.
Yeah. Also, I'm super excited to hear that you're collaborating with so many people from different expertise, because that I think makes the whole Mendel Bio platform so good. Let's say, because you're taking into account every different angle, which we all need in future biology, especially if we go to medical trials, right? We need to understand the biology, but also like the medical outcome. So this is really beautiful. So you said that the main goal is also to make synthetic biology more accessible, right? So as a last question to this topic, do you imagine a future where maybe small research labs, startups, or even universities can easily use these advanced tools without needing massive resources? Or what do you think will be the future of that?
No, definitely. Maybe one day you'll have a little printer at home that can print your medicine, or you can print your food. I don't know, or
Who
on
knows? Yeah.
on Mars. One day we'll live on Mars, according to some people. So
Yes,
so you
exactly.
need food, right? You cannot grow
Yeah,
cows or
yeah,
peas there.
yeah.
Um, so um, so definitely the aim is to democratize this thing, and it will happen.
Mm.
it will be cheap enough, it will be small enough, it will be understandable. Biology will not be, will go from life science to engineering. So yeah, I really believe that it will touch everyone's life, like firsthand.
So, guys, you are here at the forefront, so we will know if MNDL Bio in 5 years revolutionized everything. I will come back to you, Eran. So then we go to the next section where the future of synthetic biology is making biology for everyone, as we now had as a nice introduction. So as you said right now, so democratizing synthetic biology. So for many listeners, this is like a vague kind of term for them. Can you maybe just get the audience on the same page by explaining what do you actually mean with that?
Yeah, of course. 40 years ago, until 40 years ago, insulin for diabetes, it's the classical example, right?
Yes.
So
Yeah.
insulin for diabetes used to come from the pancreas of animals. It has problems. It's expensive, complicated. It's not exactly the same, like molecularly. It's not the same insulin because one is a cow's insulin and we need a human insulin, so it's not the same insulin.
Yeah.
So 40 years ago, scientists understood how to take the gene or a piece of DNA from human, which is in charge, this piece of DNA is in charge of producing insulin, and put it inside bacteria. And all of a sudden, this bacteria started to produce insulin. Again, it sounds easy, but it's not easy at all, as some of you know. But that's the first what's called recombinant protein that was out there in the commercialized. Since then, as I said, thousands of different proteins every year, including the enzymes in your detergent, in your laundry powder, or including spider silk, or many things. Thousands of different products are produced by little organisms, by bacteria or by yeasts or by and so on. So that's synthetic biology, being able to engineer organisms and to make them produce something that they don't produce naturally.
Yes.
And it is complicated.
That's for sure. And especially the spider silk is very, delicate, right? It has this really amazing properties. You can stretch it and it's so good. And I think very recently, some years ago, people actually in the lab could kind of recreate that kind of spider silk, which for me was like mind blowing that we were able to. And there's like a whole other universe where we are trying to mimic even the nature because it's so good in what it's doing, right? That we can use for technology. So that's kind of a very interesting way to see it. And how do you think can AI now make things possible to help biology, make biology more accessible? So what do you think is AI capable of when we are not talking about making weird AI videos or something? So really using AI for understanding biology.
So I'll tell you, AI is critical. AI is critical in re-engineering proteins. Okay, and you see AlphaFold, Google's AlphaFold, and all the novelty in this side. It was we needed AI to be able to engineer new novel proteins. Okay, the novel designs.
Yeah.
We are in the world of DNA. The difference here in terms of AI, difference between protein and the world of proteins and the world of DNA is that in DNA you don't have data. You don't have expression data of every mutation in DNA. as I said, there are one with 200 possibilities to make every protein, and there are hundreds of millions of proteins in the world, and maybe tens of millions of different host organisms. So there's a lot of data that is lacking. But with smart moves and with a lot of scientific collaboration and novelties, we are able to use I believe it will happen. I don't know if it will be in 5 years or in 15 years, but one day we will be able to use AI and a large language model, just like ChatGPT is able to write a new book from an idea. We'll be able to rewrite the whole genome of a new bacteria, or at least the part of a bacteria which is in charge of producing whatever protein or molecule that you are interested to produce. It will happen. We're not there yet.
So, as a context, I have a really nice friend who is more on the chemistry side, and she teamed up with also someone from the Weizmann Institute, so Sarel Fleischmann, maybe you know him,
Yes.
who developed this really cool tool called FunClip, where we actually could do like feed in the structure of an enzyme, and then depending on what you want, you can actually change the active site. And she, in her PhD, just published now, came out, she actually used these enzymes, turned them around, so they are actually even more efficient in what they're doing, in really chemicals that a chemist cannot really easily synthesize. And I think that's the power of AI, right? That we use that knowledge that we feed in to actually get really cool outcomes, to design such enzymes that we need to have. And I already, if I'm correct, I read that some of these enzymes that were discovered in bacteria that are capable of, for instance, reducing plastic, right? So they can digest plastic.
Yes.
So this is quite impressive to see if you have fine-tuned them to actually do that in large scale. It immensely helps us with the problem of the plastic crisis that we have, right? So you are at the forefront with your company. That is super exciting. And maybe on that, what do you think if we have now the AI? So some people rely heavily on AI and don't think so much anymore, right? So how do you see the role of the human creativity and the scientific institution evolving in the context of AI? So should we believe everything, or should we be skeptical, or how do you think would be
No,
the best approach?
no, of course, things are evolving. We all know that we cannot rely solely on AI, but we are going that way. I mean, it's becoming more and more reliable, more and more related and based on real facts. You know, in our context, okay, in Mendel's context, every user or every customer with which we work, we provide them with sequences. Okay, then he needs to take the sequence, synthesize them, build, and so on. And at the end of the day, he gets a certain amount of protein, which is our goal is to have as much protein as possible. So he gets a certain amount of protein for every variant, for every sequence of DNA that we gave him. Each time that he feeds back, there is a system in our platform. There is a system where you can feedback. You can provide the feedback on the expression levels. It's a little bit like on the ChatGPT when you have a good and a good
Exactly.
good answer or not a good answer. So it's about the same. These answers are critical and make our tool and also ChatGPT or any AI in the world improve with time.
Mm-hmm. Maybe a bit more provocative, but many scientists and healthcare professionals are asking the question if AI will eventually replace us, right? So my question to you is, how do you see the future of this technology? And if it actually has the potential to make us better, which has been shown by a very famous medical professional who was here in Munich, Eric Topol, was actually talking about this here. And he thinks that AI will actually help to get us back together. So I would be very crucial to me to know from you, what do you think what AI will do, especially now in your context, and if it will replace us or not?
Um, that's a good question. I want to hear why he said that AI will bring us back together. It's interesting. I'm curious to know his argument, but I think that yeah, I think that you can take any technology, you can say it about almost every technology, and take it to a bad place or take it to a good place.
Yeah.
You can say it about cars and trains and the telephone. Okay,
Yes.
so I'm not a futurist, but I believe that if you take this AI, the capabilities of AI, which are helping us a lot in many ways. There are not many people anymore that write an email without
Yeah.
having it proofed in an AI, some AI tool.
Yeah.
So and it's becoming the new norm. Okay, the new norm today is that everybody does it. So we are
Yeah.
increasing the level of written communication. Then it will be
Yes. Yeah.
increasing the level of oral communication.
Yeah.
And
Yeah.
if you take it to a good place, like we are trying to do at Mendel, and where other many companies are doing and are trying to do. I think that AI will be wonderful to all of us. Will
Mm.
make the world a
Um,
better place.
yeah. So to your question, what he said, so especially in the context of healthcare professional and patient. So taking off the workload that many MDs have to do, writing the reports and everything. And right now it's possible to actually give it to a system that records everything,
Yeah,
that they don't have to type manually and they have more time for their patients. And
yeah,
he thinks that that helps to reconnect between MD and patient to get a better relationship. So that was that. And I also think, even though there are a lot of people skeptic out there, it is, of course, good to be skeptic. But every technology that we had till now always had a positive impact on us, especially when it comes to innovations, if it comes to problem solving, right? And a very good example is also Dr. Feigenbaum, who uses AI to actually repurpose drugs in other
yeah.
contexts of diseases, right? Leveraging the good part and using it, I think that is where science and medicine is heading, that we will have AI integrated. Of course, you need to take it with a grain of salt, because every good large language model starts with a good data set. If the data set is not good, you cannot have a good large language model.
That is
So
correct.
creating that kind of good data set is crucial for a good large language model. But I guess feeding in the good data and letting it learn from the biology, I think that's where we are heading, and we should embrace this cool innovations and also support, like you, MNDL Bio, to actually get to the next level. So I think it's a really, really cool thing. And yeah, we are nearly coming to an end, but maybe before we do that, what do you think will be possible in the next 10 years, especially when you say biology speaks to everyone? What do you think, what kind of changes will you expect in medicine, food, or everyday life based on what Mendel maybe can do?
Um, what do I expect in 10 years is a long time.
Yeah.
Uh, I expect that we I think that we need to focus, that we will focus on what matters most. Technology, until, let's say, today, technology belongs, or technology is run by engineers. There are today, I think that there are something like 30 million software engineers today in the world, something like that, maybe 20, 40. These are the soon, or actually almost today already, there are not 30 million, there are hundreds of millions or even billions that are software engineers, because everyone can ask Claude or Perplexity or ChatGPT to write a code to do whatever application.
True. Yeah.
Right? So the technological barrier is becoming not a barrier anymore. And that's also what we're trying to do here with Mendel, to overcome this, to close this gap of the technology or scientific gap, which again, I believe it is impacting, and it will impact even more. All of us.
Yeah, I think so too. So we are coming to an end of our episode, Eran, and I usually give this little segment that I have here to my guest to address common misconceptions, or even sometimes if they're science communicators and they want to talk about misinformation online, which is unfortunately happening a lot. So I would like to give you the stage now to address something to the audience, or tell the audience that you would like to remember them. So the stage is yours.
I'll tell you a funny story, okay? I think 30 years ago at school. I was a kid at school, and I got a punishment from my teacher. And the punishment was because I told her I did something. I don't even remember why I got this. But the punishment, I remember this task that she asked me to do, was to write a presentation to present to the whole class about what is DNA and what is RNA. It was in class of biology. I remember it really well. And who would have thought that 25 years, 30 years later, that would be what I'm doing. All I was interested in was software. I was coding and playing with my friends and coding on a computer. That was. I think that we are living in a super interesting area. Of course, the AI is changing the whole world, of course, okay, but everybody, that's clear. But I think the biology revolution is here, is here to stay, is just beginning. I don't think that most people know what synthetic biology is. For sure, they don't know what recombinant proteins are, but it will knock on everyone's door, and it will do. I'm sure that it will do very good things to medicine, with new medicine and new therapies, and to food, and to nature, and to pollution, and so on. So,
wonderful words, and to the people, because of recombinant, because they say might not affect my life. Well, everyone you had a great example with the insulin. Everyone who is using insulin, guys, mostly of them are recombinantly made. And this is something that is life-changing. And this is also happening also in other things. My father, for instance, has an autoimmune disease, and he's getting a monoclonal antibody that has been produced, right? So this is all kind of things that are kind of already related to you from one or to the other in your life. So it's already knocking on our doors a bit. And I think in future it will be even more. So thank you very much, Eran. We have reached our conversation with Eran now. Thanks again for having your time. I know you're very busy.
Thank you.
And maybe in the future we could have you back for another groundbreaking insights, what Mendel maybe did. If you got interested in Eran's work or in general of MNDL Bio, please follow him on LinkedIn and the website. And of course, you can find every link in the description box. And of course, if you enjoyed the conversation, please make sure to like it and subscribe if you want to see more great episodes with future scientists and medical professionals. And don't forget, of course, to follow and subscribe. Eran, you know I have a slogan at the end of my podcast, and I would feel very honored if you say that with me together.
No problem.
Okay, so I start. 1, 2, 3. Let a little dose.
Little dose of honest science.
Yes. Exactly, in your life and brighten up your day.
Correct.
Yes. So, guys, thank you very much for your time, and we see us in the next episode. Till then, have a wonderful time. See you. Bye.
Daily dose of honest science.